Hey guys, let's go over this incredibly important paper published in Nature in August 2024. It's titled Fibrin Drives Thrombo-Inflammation and Neuropathology in Covid-19. This groundbreaking research builds on an existing body of work and explains how Covid-19 can alter blood clot formation, penetrate into our brains, and illustrates how our immune system's response to the disease might be the root problem for those suffering with long COVID. Okay, let's unpack this. Here's some background. Leading up to its publishing as early as 2020, reports of blood clots found in post-mortem examinations began to surface from clinicians throughout the field, even appearing in tissue samples on the microscopic level when analyzed. The researchers here aim to explain the mechanism behind these clots and the hypercoagulative state COVID seems to elicit. Quick refresh on how blood clots form. Fibrinogen is a protein secreted by the liver and always present in the blood. When an injury occurs, thrombin, an enzyme, activates fibrinogen and converts it into fibrin, which is the mesh that sticks to other blood components like platelets and red blood cells, creating a clot. The study had many parts. Here's the setup. Researchers first used computer modeling to understand which sites spike protein and fibrinogen would bind together and found several contenders, with the gamma 390-396 region being of interest. This is an important region, not only because it was a good adhesion point, but gamma 390 is known to play an important role in our body's immunity, specifically as an activator of nearby immune cells, which in other diseases like rheumatoid arthritis and multiple sclerosis can hyperactivate and lead to unwanted chronic inflammation. With the background analysis done, the experiment continued with 3 different sets of mice, all infected with the beta variant of COVID-19. What differentiated the mice was how researchers genetically altered their fibrinogen. The first set was a wild type, simply normal mice, and acted as a control. These mice have normal fibrinogen, their clotting mechanisms are preserved, and we can observe a normal immune response and resulting damage from a COVID infection. The next set is similar to the wild type, except that these mice were genetically altered so that their fibrinogen was missing the gamma 390 region. When infected, these mice can still produce fibrin clots, but by removing this gamma portion, the immune response that these clots might elicit is removed. This is important, since it allows us to see if any negative effects are coming from the clots themselves or the immune response to the fibrin clots. Let's call this set the gamma mutants. The last set are fibrinogen deficient, genetically modified to completely lack the building block of fibrin. When infected, these mice will not produce any clots and will allow us to observe an immune response in absence of a clotting mechanism. Okay, here's the first finding. Spike protein from the virus confirmed computer models and directly bound to fibrinogen, resulting in superdense, misshapen fibrin amyloid clots in both wild-type and gamma mutants. In vitro tests were also performed with human plasma. A scanning electron microscope further revealed the abnormal clot structure. Here's a normal fibrin clot in human plasma. And here's the same clot, but now in the presence of spike protein. They further tested if the altered structure would affect the breakdown of clots, known as fibrinolysis, and found that these fibrin clots, tainted with the spike protein, persisted and didn't dissolve under normal circumstances, like in the presence of plasminogen. The authors further note that these findings are consistent with the blood clots being found in COVID-19 patients. Okay, here are the next set of findings. Major deposits of fibrin clots were found in both the lungs and the brains, causing all kinds of problems. Starting with the lungs, we can see major fibrin deposits build up throughout its tissue after infection, and an equal amount of damage characterized by fibrosis or scarring in the lung. Here we can see a thick buildup of collagen along the alluvial walls of the organ. Fibrin deposits even got into the brain, building on the body of evidence that COVID can compromise the blood-brain barrier, allowing viral proteins and other components like misshapen fibrin clots to cross and cause neural dysfunction. What kind of dysfunction? Well, where the researchers found fibrin deposits in the brain, they also found a complementary loss in myelin density. Myelin is the fatty sheathing found in the axons of our neurons, and a loss in this white fatty insulation can impede with proper signal transmission. Here is the myelin density and sparse fibrin deposits of a healthy uninfected mouse. We can observe an intact and dense network of myelin. And here is our wild type, categorized by high fibrin deposits and a disturbed and deteriorated myelin density. So what was causing this damage? Was it the misshapen, dense fibrin structures themselves? Here's where having the gamma mutants is so valuable, because even though the clotting was present, there was less damage than the plain wild-type mice, suggesting what was causing the majority of the damage wasn't the fibrin clots themselves, but the immune response to the clots. And the difference in the immune response was stark. Researchers tracked several immune cells and their markers to better understand the intensity and the extent of this hyperactivation. In the lungs, we see a large difference in macrophage activity between wild-type and the gamma mutants. And where there are macrophages, there is high oxidative stress, as they release reactive oxygen species to mitigate the perceived immune threat. This 4H&E panel shows cell peroxidation, or those cells which have had their outer lipid membrane damaged as a result of high oxidative stress, with the fibrinogen deficient having the least, the wild type having the most, and the gamma mutants somewhere in between. This panel shows the clear aftermath of a hyperactive and self-harming immune system in the lungs. When taking a closer look at the brain, an adverse immune response can be observed. Here again we can see fibrin microclots crossing the blood-brain barrier and microglial immune activity being localized around them. When tracing IBA1, a protein microglia release, we can measure their hyperreactivity. When the entire brain was scanned, microglial immune activity was measured, and some important regions stood out, like the corpus callosum, hippocampus, and especially the frontal cortex. This heightened and persistent microglial activity equates to neural inflammation and can contribute to neural degeneration, leading to an observed neuronal loss after infection. Here's the last major finding of the paper, and exposes a potential negative feedback loop that may explain the persistent symptoms observed by those with long COVID. While macrophages and microglial cells were hyperactive, there was an observed drop in natural killer or NK cells, another immune cell which is vital for clearance of a virus and its proteins post-infection. Across the wild-type mice, a drastic drop of NK cells were observed, and this drop correlated with what we would expect, which is an inability for the immune system to properly process and clear out both the virus and its component parts. These next slides look at N protein, which is the nucleocapsid or the body which holds the viral RNA needed for replication and proliferation. Here we can see the result of a depleted culture of natural killer cells. Their low numbers show an obvious inability to not only clear the virus, but also the spike protein that is causing the thrombo-inflammatory effect and driving the pathology. This sets the stage for a nasty negative feedback loop, where poor viral clearance leads to the virus lingering and spreading more spike protein, in turn making more microclots which resist breakdown and leads to even more lung and brain damage via hyperactive macrophages and microglia. The combination of reduced NK cell activity, which hinders viral clearance, and fibrin clots resistant to breakdown provides a compelling explanation for the long and long COVID. This persistent viral presence and chronic inflammation, especially of the kind found in the frontal cortex, may drive the prolonged and debilitating symptoms experienced by so many suffering with the condition.